safe and efficient if the right protocols are applied. This webinar focuses on Qiagen's standardized, safe, and reliable manual and automated sample preparation protocols, which can be used for a whole range of foodstuffs, regardless of inhibitor contents. The presentation will cover a number of topics. First, discuss the importance of reliable sample preparation for food safety testing. Second, illustrate the benefits of Qiagen's modified CTAB method for DNA extraction from food. Show the process safety of DNA extraction from bacterial enrichment cultures, and finally, introduce data on effective inhibitor removal. It's now my pleasure to introduce our speaker, Dr. Marcia Armstrong. Dr. Armstrong obtained her doctorate in nutrition from the Harvard School of Public Health. After postdoctoral research fellowship in the gastroenterology department at Brigham and Women's Hospital, a teaching affiliate of Harvard Medical School, she was an assistant professor in the Department of Physiology at the University of Massachusetts Medical School. She's been with Qiagen for 20 years and is currently Global Scientific Affairs Manager with the Food Safety Testing Team. Once again, please feel free to send any questions that you may have during her presentation, and we'll address them at the end. So without further ado, I give you Dr. Armstrong.
Hello, everybody. Thank you for coming and joining our webinar, and thank you for your introduction, Keith. As is very evident in the news recently, food safety and food quality are issues of great concern. We've been hearing throughout the month of June about the E. coli 0104H4 outbreak in northern Germany, which has raised a lot of questions about food safety and food safety testing. In addition, we have other headlines, should we say, about food safety. For instance, adulteration of foods, the inclusion of, let's say, questionable or non-authenticated ingredients in the food that you are getting. For instance, people will use apricot kernels instead of almonds to make marzipan. People will add cattle or other kinds of ruminant brains to cattle feed, which is the cause of BSE in the past. We use turkey instead of pork to make sausage without declaring pork. Many foods are recalled by the FDA because of the non-declared presence of adulterants or non-food items. So all of these things tell us that food safety and food quality are great concerns in the marketplace currently and in the past and in the future. One other piece of concern, of course, is genetically modified organisms, and that is very important, particularly to export food to Europe and for the importation of food in Europe, because not only do European regulations require that GMOs be tested for, but you also have to know how much GMO is there, because there is an amount of GMO that is possible. So with all of those issues surrounding the market at this point, it's important for people to have rapid methods to detect food safety issues, food quality issues, because if you don't have rapid methods, you can't release your food quickly and easily to the market. So in this presentation, my goal is to demonstrate how Qiagen has a role in this emerging arena and how Qiagen solutions provide rapid, sensitive, and specific solutions for sample prep and for PCR-based food safety testing. Here is a quick overview, just to reiterate what Keith said a moment ago. We'll introduce a little bit about Qiagen, have an overview of Qiagen's food safety solutions, an overview of, or just descriptions of the food and pathogen DNA preparation methodologies, and also some discussion about the solutions for PCR detection in the portfolio. So Qiagen, the core of Qiagen's work over many years has been sample technologies and assay technologies. This is our core focus. Our core focus has applications in many different market areas, from molecular diagnostics, academic research, pharmaceuticals, and now we have applications in food safety and quality testing in the applied testing markets as well. Qiagen is a global company, has centers of excellence in Europe, in the United States, in the Maryland area, and also in China. We have a number of sites worldwide, so that wherever you are, we are there and available to support your work. The core of Qiagen sample and assay technologies is that you can take any complex biological sample, be that sample blood, tissue, food, etc., and from that complex biological sample you can purify the nucleic acids. In this case we're talking about DNA, obtain that DNA, and then get information about that DNA through assay technologies. So let's turn our view a little bit from the larger overview of Qiagen to taking a look at our food safety solutions. Qiagen's food safety solutions take the sample to assay technologies paradigm. We start with foods that can be very complex and that need to be disrupted, and we have solutions for disrupting, that is, for homogenizing and for lysing foods. From that, we can purify the DNA. We can set up the assays using various assay setup solutions. And from that, we can get the information from the DNA, whether it be the presence of pathogen, genetic information, GMO information, and so on. We can get that target information from the DNA that we've purified. And just a quick diversion here just to mention PCR. Certainly in the food arena, PCR is a newcomer to the detection systems in the food arena. Mostly we've been working with culture in the past, immunology, immunological assays, but here we're talking about PCR. And what does PCR do for us? Basically, it's a method for making multiple copies of DNA. You start with your purified template DNA. You then denature that. You take primers which will bind to your denatured DNA, cool that down a bit, then you extend to make copies of the DNA, and you start the cycle again. And every cycle makes geometrically increasing numbers of the template DNA or copies of the template DNA, so that at the end you have, after about 40 cycles, you have huge amounts, relatively huge amounts anyway, of DNA that you can use in your downstream application. PCR then allows you to provide rapid, sensitive, and highly specific results in a very short amount of time. Certainly one of the issues in foods is that foods need to be tested rapidly so that the food can be released to go to market. And since food is perishable, the amount of time that you have to do testing on the food is quite short. And the more rapid your methods are, the longer time your food has to be in the marketplace as opposed to waiting for testing and release. And it needs to be reliable and specific so that the answers that you get are good and you don't have to have recalls down the road. So you look here at the top line, real-time PCR uses a primary enrichment, a DNA extraction, and then detection by real-time PCR. You can do this detection and get an answer within about 3 hours after your overnight primary enrichment, so that you are ready to release your food products within about 24, 27 hours. Immunoassay has decreased specificity. You usually need to do 2 levels of enrichment, and the immunoassay takes up to a day and a half to 2 days after the enrichment to give release of the product. And traditional methods are surrounded or involve traditional culture, and the cultures are several steps. You get an answer, a negative answer meaning there is nothing there, within about 2 days, and then confirmations take another 2, 3, 4 days. So it's very labor intensive and takes a very long time. So the American food safety portfolio is in 3 parts. We have one part, which is the D&Easy American Food Extraction Kit that is involved in extraction of food DNA. There is American DNA Bacteria Kit or Bacteria Plus Kit, and these are for the extraction of pathogen DNA. And then there are real-time PCR systems for the detection of pathogens, GMO, ingredient authenticity, all done by using the purified DNA and then going into real-time PCR. So the advantage of having nucleic acid detection by PCR in the portfolio, here we have pathogens, GMO detection, and ingredient authenticity, is that we get very good performance, high sensitivity, specificity, robustness, and reproducibility, regardless of the sample type. It's one of the things we've tried to build into this portfolio is very simplified and unified methods of purification and detection. So sample type becomes not important. It's only the processing that becomes the important part. It's rapid. DNA extraction from your food can be in less than 2.5 hours for 30 samples. The time for result from extracted DNA to your PCR information is less than 90 minutes. It's user-friendly, sample preparation, harmonized detection protocols, and it's a proven innovative technology that's used in many public health and safety arenas such as forensics, biosecurity, food testing, and so on. So let's delve a little bit deeper into the American solutions for food and pathogen DNA prep. Let's talk about the D&Easy food prep first. One of the challenges with getting DNA from food, or there are many challenges with getting DNA from food because it's highly complex, and the food frequently has been highly processed. So with high process and high complexity, you get big difficulties in making DNA. So we went out to look for, or to develop a universal method for all foods. So basically one solution would fit any kind of food. We looked at solutions that are out there and looked at CTAB, feeling that that was a very good sort of classical method for obtaining DNA from food. And then we also wanted to make sure that we could recover DNA fragments because a lot of processing of food causes the DNA to become very fragmented, and there is information to be gained from fragments of DNA as long as you can recover those. The other challenges with that foods have high inhibitor contents. They could be highly processed, little processing. Some have more DNA, some have less. So trying to get a universal protocol was challenging. So we went to look at CTAB. On the left, you can see the sort of classical CTAB methodology, which starts with the CTAB inhibitor precipitation, followed by one chloroform, then a second chloroform extraction, then involving another precipitation of the DNA, an alcohol precipitation, many washes, and this whole protocol can take you as many as 2.5 days to actually get DNA to work with. And we started with that because the CTAB seemed to be a very good method to start with, and we're calling this the Qiagen second-generation CTAB DNA prep protocol. So we start with the CTAB precipitation, then do the chloroform extraction, and we take the material that's been processed so far, and we then do a further cleanup on a column. So these are regular Qiagen columns. The protocol time is 2.5 hours rather than a couple of days. You can do 30 samples in these 2.5 hours, and the prep performance is equivalent to a traditional CTAB protocol in a fraction of the time. And so here we see that basically we've gotten rid of a lot of extraction and precipitation aspects of the classical protocol, put that into the Qiagen CTAB protocol. So the food extraction protocol is a universal prep system that is equivalent to CTAB purification. The efficiency is independent of the matrix, and there's no need to split the workflow. So you don't have to have one set of samples. If you have many different samples that you want to test, you don't have to separate each kind of food and do them individually. You can do all different food types in a single run. It's the efficient separation from the inhibitors, and you can recover fragments of DNA from processed food, and it is automatable, or at least partially automatable, either with a QIAcube or potentially with a QIAextractor, which is a 96-sample throughput instrument. Okay, we have tested a wide variety of foods to see that it works, and on the right you can see a list of the kinds of foods that we have tested in our laboratory, from ketchup to olive oil. In between we have chocolate, different kinds of chocolate, cocoa, cornflakes, which are very difficult, milk, and things like this. So foods that are of great interest and can be very difficult. Foods are fatty foods, foods with strong inhibitors, highly processed foods, foods with low DNA content, and also high DNA content. And all of these work with the Qiagen food extraction protocol. And here's a little bit of PCR data. This is actually a spiking study where we took a PCR template that we had already made and put it into the food, and then did a PCR on that particular DNA template to see how it worked. What you would expect to see if you have undiluted sample and then diluted at 1 to 10 is a shift to the right of about 3 CT units. And this is what you see in all of these. Chocolate on the top, which is strongly inhibitory, ketchup in the middle, which is highly processed with low DNA content, and on the bottom we have cornflakes, which again is a highly processed food with low DNA content. And the fact that the food is strongly swelling is a challenge in the purification. In all of these cases, we have we can show that the undiluted, conversely, that the 1 to 10 dilution, dilute the food, gives you a shift of about 3 CT units to the right, which is what you'd expect for an uninhibited sample. Another study we did on the same thing was taking two fairly complex matrices, peanut butter with the blue bars and chocolate on the bottom with the sort of pinkish reddish bars. And looking at the Maricon, did a normalized CT study and then compared that to sample preps from some other manufacturers. And certainly the Maricon, and this is the same starting material, so that the only influence is the different sample prep. And the Maricon in both cases, both the peanut butter and chocolate, shows you a lower CT, which suggests that there is less inhibition or better removal of the inhibitors from using the Maricon food DNA extraction kit. Did a further study where we wanted to see what the difference was with a 1 to 10 dilution. Did we get that 3 CT shift to the right, or did we see something else happen? With the Maricon, you can see that both with chocolate and peanut butter, you have a 3 CT shift, which would be a plus shift to the right. With the other 3 sample preps, there is a slight shift with peanut butter and all 3 different sample preps, but with chocolate, especially from 2 of the different sample prep types, there is a shift to the left when you do a 1 to 10 dilution, which suggests that the initial undiluted sample was highly inhibited. So it gives further credence to the fact that the inhibitors have been removed by the Maricon food DNA processing. And here is one further study done with the Maricon food DNA kit. We talked about the fact that that certain foods are highly processed and that you get small fragments produced. So our standard protocol, which starts with 2 grams of food in the processing, gives you a particular, this is again a spiking experiment, and then we compared that to the small fragment protocol. And we find that with the small fragment protocol, we actually get more, which we'd expect, because the small fragment protocol allows you to recover more DNA, small DNA that's still important in the PCR, and you get a small shift to the left, which indicates that you have more sensitive PCR with the small fragment protocol. Cornflakes is a perfect example of a food where you get fragmentation of the DNA in the processing. Cocoa and ketchup don't show as much difference because they aren't as highly processed and don't have as much shift to the left. Okay, so let's move to DNA extraction from for food pathogens from cultures. There are a number of different, obviously, a number of different bacterial pathogens that are of great interest in the market today. Salmonella certainly is responsible for many, many cases of foodborne illness. Listeria monocytogenes is perhaps lesser amounts of foodborne illness, but it's a terrible disease and can be quite, the mortality from L. mono infections is quite high. And there's Chronobacter, which is interesting for in infant formula, and Campylobacter, which is actually, again, probably one of the largest foodborne illnesses out there. It doesn't cause a huge amount of disease, of morbidity, but it certainly has lots of a lot of foodborne illnesses caused by Campylobacter. So what we've done is taken a look at these different, particularly with these very common and very important pathogens, and we have a thermolysis kit for gram-negative bacteria and a mechanical lysis protocol for gram-positive. You need to break those gram-positive walls, so there's a treatment with glass bead lysis as well. So let me just go through this a little bit and show you the protocols. Basically, all the assays for bacteria start with an overnight culture. You take the enrichment culture, which is indicated by the hand with the tube, and then you either do the gram-negative bacteria or the gram-positive bacteria, depending what your starting bacteria is and what you're looking for. Both kits provide lysis buffer systems, either thermal or mechanical, additives for DNA stabilization, additives for inhibitor removal, and provide you with standardized and efficient sample prep. So these are the workflows. On the left, we have the mechanical lysis. You take a sample from your culture broth, spin the cells out, and these cells are going to contain the bacteria. You resuspend them in a lysis buffer, and you transfer that to a bead tube, and you bead it either in a vortex or in some kind of mechanical disruptor machine. Spin that again, and the supernatant is going to contain the DNA for your PCR. The thermolysis is even simpler. You take the sample from your culture broth, you spin out the cells, you resuspend the cell pellet in lysis buffer, keep the sample for 10 minutes, spin it again, and the supernatant is ready for being used in your PCR. This slide just demonstrates the difference between the mechanical and thermolysis protocols for Listeria. This is our gram-positive bacteria, and you get certainly slightly enhanced PCR recoveries or PCR reactions with the certainly with low, undiluted and lower dilutions of the Listeria. So it does make a difference. If you use mechanical lysis, you get better PCR performance with the gram-positive bacteria. Okay, so let's turn our attention now to the Maricon solutions for PCR detection. And this is the portfolio that we have available at the moment. Pathogens, we have a wide variety of pathogens that we can detect, from Salmonella, L. mono, Campylobacter triple. So these are the 3 Campylobacter species that are important in illness, and there's a Campylobacter species which detects several additional Campylobacter species. There is a detection for verotoxin-producing E. coli. So this actually will assess step 1 and step 2, the 2 verotoxins in E. coli. Have a system for Staph aureus, Yersinia, Shigella species, Chronobacter sakazaki, Legionella, and Legionella pneumophilia. So a wide variety of pathogen detection assays. We have some for GMO detection, some screening, one looking at Roundup Ready soy, several for, again, a variety for various screens, and then 2 quantitative assays, one quantitative Roundup Ready soy and quantitative MON 810. And this is because European regulations require that you prove that there is no GMO, or if there is GMO, that you have to quantitate how much is there. If there are some certain, it's like 0.9% is allowed. So for anyone who is exporting food from the U.S. or from anywhere to Europe or Japan, it's important to know whether you have GMO in your food, and important to know how much is there in order to sell the food. I'll also have a number of tests for ingredient authenticity, pork, soy, cattle, chicken, turkey, corn, and apricot kernels of use in marzipan manufacture. And we will have a panel of allergen detection kits in 2012. So then the thing that we then needed to look was have these assays, and the assays are generally primer probes. But what kind of master mix do you use? And what we have found over the years in our research is that the master mix is a very important factor in working with samples that can be highly inhibited. So we did some studies where we looked at inhibitor concentrations with the number of template concentrations, trying to understand exactly which of our master mixes would be the ones that would work best in the PCR assays for in the food portfolio. These are the assay chemistries that we added to our master mix. So a number that you would find in DNA purification. So these sort of are residuals, SDS, CTAF, sodium chloride, and so on. Neutral polysaccharides and acidic polysaccharides, which are used in the manufacture of food for various, can be used to replace sugar, fat, flour, enhance taste and texture as gels, stabilizers, and so on. Polyphenols are very important, certainly in things like cocoa, wine, fruits, vegetables, chocolate, because polyphenols is a class of compounds that appear in these foods and make it very difficult to do PCR. And then certainly there are proteins that are added to foods as well. So these were the wide variety of chemicals that were added to the PCR master mix to try to understand which one of our master mixes and whether our master mix was in fact tolerant to the presence of inhibitors. And this is the kind of information that we got. We looked for the effects on the general CT range, the quenching of the N fluorescence, scattering of N fluorescence, because of course all of these PCRs use a fluorescent probe at the end, and if anything interferes with the fluorescence of that probe, then you can have off effects. Buffer pH can be important. The replicates can scatter because of the presence of inhibitors, and then it can also boost the PCR effects. So we evaluated the various master mixes and came up with the fact that our multiplex PCR master mix was the one that performed the best with the most inhibitors. And you can see the Americon master mix with the blue bars versus the other supplier, which is this pinky purpley red bars. And the CT change with the addition of the inhibitors for the Americon master mix was negligible. For the tests that were done with multiplex PCR master mix, but quite significant from other manufacturers. So we then took this information to design the kits. Our kits come in 24 reactions or 96 reaction size. There is the Americon assay, which is the primer probe for the target, and the internal amplification probe. There is a positive control DNA specific for the particular test, and then the multiplex PCR master mix, which is the fast-cycling hot StarTac plus polymerase that we found to be highly tolerant towards the inhibitors. We also have some kit components with dilution buffers, ROX dye, should you want to use these assays with a cycler that requires ROX. Certainly the Qiagen Rotogene Q does not require ROX. And then RNase-free water for you to dilute your samples. One of the key pieces about the PCR assay portfolio is that every assay within the entire range of assays is harmonized. So if you want to do if you should want to do an ingredient authenticity for soy versus a Roundup Ready soy for the GMO versus Salmonella, they could all be run in the same PCR reaction, in the same cycle. So you could put them all into the PCR cycler at once, and you could run all three of them simultaneously and get the answers. So the PCR volumes are 20 microliters, 10 of template, 10 of assay. There is an activation step of 5 minutes, and the harmonized protocol is 3 steps: denature at 15 seconds, anneal at 15 seconds, and extend at 10 seconds. There are 40 cycles for the pathogen test and 45 for the GMO. So this is a major advantage in the workflow setup and in combining analyses from different segments in a single run. So where are we going to take this now? We have a manual DNA prep from bacteria. We have manual and semi-automated DNA from food, and then we have a portfolio of tests. What we're working on currently is to fully automate the bacterial DNA extraction protocols using the Qiagen, which is a bead-based instrument that has 96 sample throughput. Ultimately, some manual high-throughput protocols that would allow you to do 96 wells of the short manual protocols, and some expansion of the assay portfolio in 2012. And just very quickly and briefly here, the Qiagen RGQ pathogen protocol uses the same workflow. You homogenize, you incubate, you take a sample, and you put the sample in the Qiagen, which is the instrument picture here. The Qiagen will take the sample, it will purify the sample, it will lyse the sample with a thermal lysis, and then use a bead-based assay to purify the DNA. And then if you have the assay setup module as well, it will set up the PCR for you, allowing you to have completely hands-off purification and assay setup in one instrument. And then you put it into your PCR cycler, and you develop, and you can get the CT. So it's very, very simple, and it allows you very nicely to have an automated system. And this is just a little bit of the data, an example of data from the Qiagen RGQ pathogen protocol. And these are 2 chocolate matrices, which are very difficult. So the one on the left is chocolate with a coffee filling, dilution series, a very nice dilution series, very even distribution of the CTs with dilution. And this is very difficult because not only do you have chocolate here, but you also have coffee, so you have a lot of inhibitors present. And then we also have milk chocolate dilution series as well. I believe this should be the last one, so thank you so much for listening. The Qiagen solutions go from sample technologies to assay technologies, and this has been applied to the